Hi everyone, I'm Sara and I'm working at the BBB laboratory, or LBH, at the University of Artois in the north of France, and this under the supervision of Dr. Maxime Culot. Within the IN3 project, I focus on the blood-brain barrier, and so here I will be presenting my results from a BBB model derived from iPSC for repeated dose toxicity testing. So let's start at the beginning. What is a BBB actually? The BBB is a specialized form of endothelial cells in close proximity of parasites, astrocytes, and other cells of the neurovascular unit. The human brain contains 650 kilometers of these capillaries, which counts for a surface area of 20 square meters. The BBB is a physical and metabolic barrier. Because of the presence of the tight junctions, passage through the paracellular pathway is excluded. And also passage through the transcellular pathway is limited because of the presence of drug metabolizing enzymes like CYPs, which can lead to degradation, and also the presence of efflux transporters like PGP and BCRP, which can pump compounds back into the bloodstream. The BBB is also a selective barrier. Only a limited amount of compounds can pass through by passive diffusion, like for example caffeine. And some nutrients or compounds are specifically transported, like for example glucose transported by the GLUT1 transporter. So why are we modeling the BBB in vitro? First of all, if you want to assess neurotoxicity, you'd actually need to know if a compound can actually reach the brain. And as a BBB controls the access from compounds to its cerebral tissue, this is very important information. Within the IN3 project, Carolina is working on a model from human 3D brain derived from iPSC. Besides that the BBB controls the access of compounds to cerebral tissue, BBB also maintains the central nervous system homeostasis. So if a compound would interact with the BBB and compromise its functionality, this could lead to indirect neurotoxicity. Let's keep in mind that we're using models, and all models are wrong, but some are useful. So we will be evaluating our BBB model on 3 important characteristics for us, and you will see the information on that later. We tried multiple different models, differentiation protocols, and in the end we decided to use the differentiation protocol from Kian et al., which was published in 2017, because this was a completely chemically defined protocol, so we didn't need to use any serum. As we're using another cell line than was reported in the Kian protocol, we also needed to do some optimizations. And this started by testing different seeding densities at this first time point. We called these different seeding densities low, middle, and high. And so after differentiation of the cells, we evaluated them for multiple characteristics at the end of the differentiation at day 10. At day 8, these cells are replated on inserts, but we also replated them in 96 well plates. So first of all, we wanted to know if these cells were really forming a barrier. And for that, we used tear measurements, transendothelial electrical resistance, and we saw that only for the lowest seeding density, there was an increase at the end of the differentiation. Then we wanted to understand if actually functional efflux transporters were present. For this, we seeded the cells in 96 well plates. We incubated them with rhodamine, which is a substrate for PGP and BCRP, for 2 hours. Afterwards, we washed the cells and we measured the fluorescence in the buffer above the cells to see the extrusion of rhodamine in the buffer. We also measured this in presence of inhibitors like verapamil and elicredar. And if these functional efflux transporters are present, then in the case with inhibitors, the fluorescent extrusion is lower. We measured this for 3 different seeding densities, and we saw that only in the presence of inhibitors at the lowest seeding density, there was a reduction seen in rhodamine. So for the lowest seeding density, we found that there was a restrictive paracellular permeability and the presence of functional efflux transporters. Then we also wanted to know if there were any endothelial markers or brain-like endothelial markers present. And so we did some stainings, for example, for the tight junction protein claudin 5, but also for vascular endothelial cadherin. So this model until now ticked our three major boxes that needed to be present. And then we started to test this model on a more regular basis. And while using it, we also realized that we should reduce the day in a specific medium from 4 days to 3 days. And when performing these differentiations in parallel, we still see that there were quite some variations in tier at the latest time point. Then we wanted to know if we could use this model for repeated dose toxicity testing. But if we want to use this model for repeated dose toxicity testing, we wanted to know if these brain-like endothelial cells retained their BBB characteristics over time. So that's the first thing that we investigated. We differentiated the cells, which the differentiation ended at day 0. And then afterwards, we kept them for 15 days and looked at the tier over 15 days, and we saw that at day 15, the tier was still higher than 500 ohm centimeters square. And we also saw the presence of claudin 5 staining or this tight junction protein at day 15. Then so we also wanted to know if these functional efflux transporters were still present, which we measured at day 0 and day 14. And we saw in the presence of these inhibitors, elacridar and verapamil, at both time points, the rhodamine extrusion was reduced. We decided to use cyclosporine A, which is an immunosuppressant, as a compound to study for repeated dose toxicity testing. This because of cyclosporine A already multiple times being studied in other cellular models. So if we wanted to assess the repeated dose toxicity, we first wanted to choose a concentration and for which we did an acute exposure. So we exposed the cells to cyclosporine A and measured the resoriphin, or we did a resazurin assay, and we only saw an influence at the 20 micromolar concentration. So we decided to use 5 micromolar, which is a therapeutic concentration, and 50 micromolar, which is a supratherapeutic concentration. So we differentiated the cells, and at day 0, we started treating the cells by changing the medium every 48 hours with 5 micromolar cyclosporine A, 50 micromolar cyclosporine A, or just none of it as a control. We measured the tier again over time, and we saw that the tier at day 1 immediately decreased. And this became significant for both conditions at day 5, and it kept on decreasing over time. We also wanted to know we also wanted to investigate the cellular stress, for which we used a lactate assay. So we measured the lactate in the medium at different time points, and we immediately saw a significant increase in lactate at the 2 different time points. There's one thing which is kind of particular here. We saw a higher increase in lactate at 5 micromolar cyclosporine A compared to 50 micromolar cyclosporine A, while you would expect that a higher concentration would lead to more cellular stress. And as therefore, we performed also a resazurin assay at day 7, and we saw a reduction in the 50 micromolar treatment condition, which was not the case in the 5 micromolar treatment condition. So possibly this could be explained by cellular loss. And this cellular loss was confirmed by staining at day of treatment 15. Besides that, we also did targeted RNA sequencing, or TempoSeq. And after getting the results from the sequencing, we used the bioconductor package DSEQ2 to assess the differentially expressed genes. We first wanted to more understand what is happening in the model without treatment. So we decided to look at the differentially expressed genes of day 1 compared to day 0, day 3 compared to day 0, 5 to 0, and so on. And we saw that the amount of differentially expressed genes is increasing over time. So there are still a lot of cellular processes changing over time in this model. So that's something to take into account, and to be aware of when using this model for repeated dose toxicity testing. Then we went to investigate the influence of cyclosporine Aas at the two concentrations. And here we compared, for example, the 5 micromolar cyclosporine Aas condition at day of treatment 1 to the controlled condition at day of treatment 1. So we always compared to the controlled condition of that specific time point. For 5 micromolar cyclosporine Aas, we saw that the amount of differentially expressed genes was increasing. And when comparing day of treatment 1 for 5 micromolar cyclosporine Aas and 50 micromolar cyclosporine Aas, we also saw a concentration-dependent increase in differentially expressed genes. And also for 50 micromolar cyclosporine Aas, the amount of differentially expressed genes was increasing over time. We excluded this last time point because there was just too much cellular loss. So then we wanted to understand which pathways were actually activated in the cells, what was happening. And for this, we used Z-scoring pathway analysis. The Z-score allows us to see the changes of the genes inside a pathway compared to the changes of the genes outside of a pathway. If a Z-score is higher than 1.96, then it's significantly overrepresented. So within a list of genes that we had for TempoSeq, we annotated them to specific stress response pathways, and then we could investigate the overrepresentation of these stress response pathways. We did this for all our time points and conditions, and we saw, for example, that the ATF4 pathway was overrepresented at all time points and the both concentrations. This XBP1, NRF2, and P53 were mainly overrepresented at the 50 micromolar treatment condition. And this, the P53 overrepresentation, could also be confirmed by the fact that we saw a lot of cellular loss. These results were also reported in other cellular models like RPTEC and TERT1 and EPRG cells. So in conclusion, the BBB characteristics were retained over time, but we still saw a lot of differentially expressed genes, meaning that there are a lot of cellular models still changing. But when we used Z-score pathway analysis, we were able to identify stress response pathways, which were also identified before in other models. So this is a very promising tool for repeated dose toxicity testing. So all models are wrong, but then the question is, how wrong do they have to be not to be useful? And then I would like to thank everybody from the IN3 project and also the people from the LBHE or the BBB laboratory in France. And thank you for watching.